Well, hi there. Welcome back. And we're going to go into part 3 of metabolism. In an overview of metabolism, just kind of review a few things. Chemical reactions in metabolism occur in metabolic pathways. Each step of that pathway is going to be coordinated or carried out by a specific enzyme, and multiple enzymes carry out multiple steps in a metabolic pathway. Metabolism can occur in two different basic types of pathways. The first are catabolic, the second are anabolic. Catabolic pathways are used to release energy that can be used to drive anabolic pathways. Remember, this is kind of a circular process. One pathway feeds the other. Catabolic pathways release energy through the breakdown of molecules. So they'll break the chemical bonds between two elements in a molecule and release the potential energy so that it can be consumed as kinetic energy. These reactions are exergonic and usually result in a negative delta G. Anabolic pathways utilize energy. They're used for the biosynthesis of organic molecules and macromolecules used by the cell. Most of their reactions are going to be endergonic, and the endergonic reactions will be coupled to exergonic reactions. And so this actual process is known as a coupled reaction, where the energy from an exergonic reaction is tied to the biosynthesis of another molecule, which is an endergonic reaction. In metabolic pathways, remember we start with our initial substrate, and that is going to enter into a reaction with the first enzyme in the pathway. That enzyme will cause a reaction to occur, which then will bring our create intermediate 1 as the product. Intermediate 1 as a product will become the reactant for enzyme 2. The product of enzyme 2 becomes the reactant for enzyme 3, and so on until we get to the end of the pathway. The products of one reaction are going to become the reactants of the next. In catabolic reactions, remember we break down things, so this is where the reactants are going to be broken down. These reactions are used in recycling properties for cells. They're going to need to recycle, such as old mitochondria, old cellular components, or proteins. Those are going to be broken down, and the energy released from those chemical bonds, like the peptide bonds between amino acids and such, the energy that's released from breaking those bonds is going to be used in endergonic reactions to create new proteins. Examples are RNA being broken down by ribonucleases, proteins being broken down by proteases, lipids being broken down by lipases, etc. The energy that is the energy that is released in these catabolic reactions can be used in the form of ATP. It can be used to form what are known as energy intermediates, and it can also be used directly as a form of energy. Now, ATP, adenosine triphosphate, we know is the primary energy molecule of the cell. It is an adenosine nucleotide with 3 phosphate groups attached to it. And that third phosphate group on the end of the ATP molecule is where majority of the energy is released during ATP phosphorylation. But in order to use ATP, the cells have to be able to make ATP. And ATP, being the major currency, energy currency of the cell, is made in 2 basic ways. First, we have what's known as substrate level phosphorylation. The second is known as chemiosmosis. You do need to know the definition of each of these and where each of these types of ATP synthesis occur in primary metabolism. So in substrate level phosphorylation, an enzyme in a metabolic pathway is going to directly transfer a phosphate from whatever its substrate is, whatever its reactant is, directly to adenosine diphosphate. So ADP plus a free phosphate group will become adenosine triphosphate. I'm going to try using this pen here so we can. So I have ADP plus a free phosphate, that's what PI is. When these react, it becomes adenosine triphosphate, or ATP. Enzyme direct transfer is going to occur in primary metabolism in two places. It will occur during glycolysis, and it will occur in the what's known as the Krebs cycle. The Krebs cycle is also referred to as the tricarboxylic or TCA cycle. The second form of ATP production is chemiosmosis. And chemiosmosis is where we take an electrochemical gradient. If we have a gradient, that means we have a membrane. So we have an electrochemical gradient, and that gradient will be used to drive diffusion. So we're going to have two different types of transport occurring here. Basically, this is happening in the electron transport chain. And the electron transport chain is the very last step in cellular respiration. We will go through each of these: glycolysis, Krebs cycle, and electron transport chain. We will go through each of these in order, and you can see where ATP production is going to be occurring in each of these steps. But to give you a general idea of chemiosmosis, in chemiosmosis, or the electron transport chain, we have a membrane. Now this is going to be occurring in the inner membrane of the mitochondria. That's where in eukaryotic cells, that is where chemiosmosis occurs. And across, oops, I erased it, sorry. So across this membrane, just do it like that, makes it a little easier. Across the membrane are several proteins. Okay, these are these big proteins. There's a small one right here. We have this one right here. And across this membrane, I'm going to change colors here to make this a little easier. I guess not. Okay. All right, so when we move through this membrane, some of our energy intermediates, on the previous slide, we saw what's called NADH. This is an energy intermediate. And this energy intermediate, what it has is it has an extra proton. Okay. And what it's going to do is it's going to come over here and it's going to drop off a hydrogen atom, or a proton. So I'm just going to make that a little plus sign. And what these proteins here do is they're proton pumps. And they're going to take that proton and they're going to pump it across the membrane. The electron from this hydrogen atom is going to be transferred from this guy here. This protein here is going to transfer it over to here. So here I have an electron. And every time this electron moves over from one protein to the next, it releases energy. And every time it releases energy, these proteins can pump another hydrogen ion across the membrane. So that what I end up building is a whole bunch of positive charges on one side of the membrane. Later on, over here is another protein known as ATP synthase. And ATP synthase allows the diffusion of protons back across the membrane and uses the energy from that diffusion to create ATP. So what we really have here is energy intermediates dropping off hydrogen atoms, or dropping off electrons and protons, using active transport. Remember when we learned about transport in the last unit? We're going to use active transport to create this electrochemical gradient, right? So we have this high charge, this high ion charge on this side over here of the membrane, and then ATP synthase is allowing diffusion, which is passive transport, allowing that diffusion back across the membrane. So we're moving against the concentration gradient here and then with the concentration gradient here, each time releasing energy that is used to drive the chemiosmotic process. Now at the end of this electron transport chain, waiting over here is an oxygen atom that is going to accept two of those electrons, right? So these are negatives, and this creates a water molecule. So we end up with a water molecule at the end of it. We will go into greater detail on this in the next lecture, but just wanted to give you a general idea of how the electrochemical gradient works in the electron transport chain. And so energy chemiosmosis occurs in the ETC, okay, in the electron transport chain. Now, how do all of these reactions work? We're talking about this movement of protons and movement of electrons, and all of this is known as a redox reaction. A redox reaction is a combination of oxidation and reduction. Oxidation is when electrons are removed. So when a molecule has electrons removed from it, it is oxidized. When the electrons are added to another molecule, that molecule will be reduced. Now, since electrons are such high energy subatomic particles, they must be bound to something. They're not allowed to just roam around the cell freely. They will kind of bump into everything and create lots of different problems and react with things. So they always have to be kind of on a leash or attached or part of a molecule. So these two reactions will always occur together. If one molecule is being oxidized, another one is being reduced. In the example of glycolysis, in glycolysis, we looked at, let me go back here. In glycolysis here, we looked at substrate level phosphorylation. And in this process, it's very similar. What we're seeing is bonds will be broken, and when they get broken, electrons get freed. Those electrons are going to be accepted by an energy carrier, NAD+, and when it accepts it, it'll become NADH. Okay. So here in these redox reactions, we are oxidizing glucose in primary metabolism in order to get electrons from it. We want these electrons because we want our energy intermediates to take them to the electron transport chain. A redox reaction is just simply an electron being moved from one molecule to another. One is being reduced by accepting the electron, one is being oxidized by losing an electron. By the way, just to clarify, it's called reduction when you're accepting an electron because the charge of the molecule will be reduced by however many electrons are being accepted, because electrons have a negative charge. So in this example here, just going through redox again, where A has been oxidized, its electron has been removed, and that electron here is being accepted by B. So A has been oxidized, but it is called the reducing agent because A is reducing B. B is oxidizing A. So A gets oxidized and B gets reduced. So that brings us to those energy intermediates I was talking about. This whole electron movement, it's occurring in glycolysis and the Krebs cycle. And what's really happening in metabolism, particularly in what we refer to as primary metabolism, the breakdown of glucose, what we're really trying to do is to rip as many electrons off of a glucose molecule as we possibly can. Because the electron transport chain, that chemiosmosis that we talked about a little bit earlier, creates far more ATP per glucose molecule than substrate level phosphorylation. So it's advantageous, or it's more effective for the cell to use electrons, pull electrons off of glucose and send those electrons to the electron transport chain. In this case, NAD+ and FADH+ are the two electron carriers. These are the guys that are running around the cell, picking up electrons, and then running those electrons over to the electron transport chain and dropping them off. So NAD+, let's just follow one here. NAD+ in glycolysis will get reduced and become NADH. Okay, so this is reduction. And then NADH will go to the electron transport chain and be oxidized. To again become NAD+, and then of course its hydrogen goes to the electron transport chain. This NAD+ is going to go back over to glycolysis or the Krebs cycle, it can be either one, and pick up another electron and get oxidized by the electron transport chain, and then go back to glycolysis or the Krebs cycle, get reduced, carry that electron to the electron transport chain, and then be oxidized. So ultimately, NADH is really its own redox reaction. This same process will happen with another energy carrier called FADH. And FADH does the exact same thing. It just drops off its electron in a different place in the electron transport chain. They both go to the electron transport chain, just where they drop off their electrons will be a little bit different. In anabolic reactions, we are synthesizing molecules. And in biosynthesis, we're going to make those big organic macromolecules that the cell needs, such as putting together molecules for nucleotides or amino acids, or even just taking individual amino acids and putting them together as a protein. When we build proteins, that's done through what's known as the translation process, and ribosomes are going to use lots of ATP. It takes 2 ATP to join 2 amino acids together to start making proteins. So we need a lot of ATP for these biosynthetic pathways. But this means that metabolic pathways have to be regulated. And gene regulation occurs in a multitude of different ways. First, we have, of course, regulation at the genetic level. DNA can be turned on or off. Enzymes are a class of proteins, which means that the genes that encode for them are found within the DNA. So we can regulate the DNA to either turn the gene on to make the protein or turn the gene off to not make the protein. That would be done through cell signaling. So like just in Unit 2, when we looked at cellular responses and how we went through these cell signaling pathways, and ultimately the signaling pathway told the nucleus to tell a gene to either make something or turn something off. That's genetic regulation. We can also have cellular regulation. Cell signaling pathways, such as hormones, will start activating proteins that turn other parts of cells on and off. They don't necessarily always have to go straight to the DNA for production. Sometimes those proteins are just sitting in the cell waiting to be activated. The last is biochemical regulation. And in biochemical regulation, pathways are actually regulating themselves. And that's what we're going to talk a little bit about, because a lot of medications and drugs work through biochemical regulation of metabolic pathways. So in biochemical regulation, we have 2 types of what we call inhibitors. We have competitive inhibitors. Competitive inhibitors do exactly what the name of it implies. They compete. So here we have foreign molecules that come in and will outcompete the substrate for an enzyme. Now this could be within a metabolic pathway or some other pathway, but at some point that enzyme will be inhibited because the competitor will attach to the enzyme, but won't actually allow, the enzyme can't carry out its true reaction simply because the substrate or the molecule that's competing, the competitive inhibitor, is not the true substrate for the enzyme. So in competitive inhibition, molecules will outcompete the substrate. One thing to note about competitive inhibitors is, particularly in pharmaceutical industry, competitive inhibitors will be metabolized. So these inhibitors, you have to watch the dosage, because competitive inhibitors work by outcompeting the substrate. And if that's the case, if your concentration of competitive inhibitor falls too low, then your medication is not going to work anymore. In non-competitive inhibitors, our inhibitor molecule will bind anywhere except for the active site of the enzyme itself. Now it does this because when you attach something or bind something to a protein, the protein will change shape. So if the protein changes shape, then the active site will no longer fit the substrate that it would normally bind to. This is known as allosteric binding. So in this case, our inhibitor doesn't have to compete with the substrate. We're not worried about it's not as concentration dependent as a competitive inhibitor would be. This allosteric site is found anywhere on the enzyme, and the protein could have multiple allosteric sites. And many cells will use non-competitive inhibitors as part of their own pathway regulation. And in this process, this is known as feedback inhibition. In feedback inhibition, the very final product of a pathway is the actual inhibitor of the pathway to begin with. And here's how this works. Down here at the bottom, we have a feedback inhibition loop. So here's enzyme 1, and our initial substrate binds into the active site, right? That's what's going here. And this pathway carries out its process, becomes intermediate 1. Intermediate 1 is the reactant or substrate for enzyme 2. Enzyme 2 carries out its reaction to make intermediate 2, which is the reactant for enzyme 3. And here we have the final product of our pathway. This final product will be used by the cell for whatever this pathway is making. So lots of it's going to go out into the cell to make whatever it is it's supposed to make. Let's say this is an amino acid. In bacteria, this pathway here is done with tryptophan, so which is an amino acid for proteins. So here we have our amino acid, and our cell is using the amino acid, making lots of proteins, but this pathway is uninhibited, which means this pathway just continues to make more and more of this particular amino acid. We'll just put AA here. Once the cell uses as much of this amino acid as it needs to use, and let's say it doesn't need any more for a short period of time, this amino acid is going to start building up in concentration inside of the cell. And when this occurs, this amino acid will allosterically bind to enzyme 1. When it allosterically binds to enzyme 1, that active site is no longer able to fit the initial substrate, and this pathway will be shut down. We will not make any of this. Now eventually, once the cell needs this amino acid again, it will start using up the free amino acid, and then also it will have greater affinity for this amino acid. It will pull it off of enzyme 1, and the pathway will start all over again. And then the pathway will now move forward again. Same thing will happen. Start building up all this amino acid. When the concentration gets high enough, the amino acid becomes the inhibitor of the pathway itself. So when a metabolic pathway, when the final product of a metabolic pathway becomes the non-competitive inhibitor of the actual pathway itself, no matter where in the pathway it is, this is known as a form of feedback inhibition. In recycling, genetic material gets broken down. This is done by a class of enzymes known as nucleases. They are going to break the nucleotides off of DNA or RNA, and they'll do this from one end of the molecule to the other. Proteins are broken down by proteases, and eukaryotic cells use a specific structure called a proteasome to break down proteins. So the proteasome is a kind of like a thing. I think of it as a room. It's just kind of a little room or closet inside of the cell in which proteins are guided into when they're no longer functioning, and the proteasome will then break them down. The proteases are located inside of the proteasome, and this is done to protect functional proteins from those protease enzymes. So here is the proteasome. The process of protein degradation is known as ubiquitinylation. Here it is spelled out right here. The proteasome itself is 4 protein rings that are stacked one on top of the other with 2 protein end caps. Now these end caps are what regulate what proteins enter and what proteins exit. So here we have a protein entering into this end of the proteasome. The proteolytic enzymes are found inside this structure right here. And then on this side, you can see our protein has been broken up into multiple pieces, or even into individual amino acids. Proteins that are broken down are those that are worn out. Maybe there are proteins that don't need to be used anymore. They've carried out their function and the cell doesn't need them anymore. And sometimes proteins don't fold properly. So if the protein doesn't fold properly, it's now non-functional. It's going to be tagged for destruction. Proteasome will only break down proteins that have been tagged with a molecule called ubiquitin. So when the cell notices or is signaled that a protein is no longer functioning, the cell will attach a ubiquitin molecule to the protein, usually in multiple areas. And here they're showing you the ubiquitin right here. And when the ubiquitin is tagged, the proteasome will then bind it and start taking in that particular protein. The enzymes that are responsible for attaching ubiquitin recognize proteins that are wrong, that are not functional or not working properly. And this allows the cell to respond very, very quickly to cellular changes That way the cell can break down one protein while building another protein and keep recycling all of those units, those amino acid units. Autophagy is another form of molecule recycling, and this is where organelles are autophaged, which is self-eating, so by the lysosome. So here we have one organelle, the lysosome, that is going to start consuming other organelles that are found inside of the cell itself. So this is not phagocytosis. Phagocytosis would be taking elements from the outside of the cell. Here, this is from the inside of the cell. It's going to do this to break down organelles that are not needed, that are not functioning properly. Sometimes organelles start wearing out. They may be damaged, things like that. It can also be used to destroy intracellular bacteria or viruses that have endangered the cell. It can be used in protein degradation if there's a multitude of proteins that need to be destroyed all at once. And the autophagosome, this is misspelled here, there should be a G here. The autophagosome is formed when we need a double membrane for this function. So cellular respiration is the way that we make ATP. And it's the process which living cells get energy from organic molecules. So when we eat our food and we break down glucose and other proteins and lipids and those sorts of things, all of those metabolic pathways all ultimately lead to cellular respiration. And glucose metabolism is, of course, number one. Now, the primary aim, the main goal of cellular respiration is to make ATP and to make energy intermediates. The three energy intermediates, NADH, FADH2, we've already talked about. There's another one called NADPH, and NADPH is used in plants. Animal cells use NADH and FADH2. Plants use all three. Aerobic respiration is the use of oxygen to create ATP. And we'll talk about how oxygen is used to do that. So in cellular respiration, we have this chemical formula down here that you need to know. It's a pretty straightforward one, pretty simple. And you will need to know that for your exam. So make sure you know the chemical formula for cellular respiration. Now, we're going to expand on ATP production. Remember, we talked about substrate level phosphorylation. We also talked about oxidative phosphorylation, which is chemiosmosis. Think about it, oxidative, because of those redox reactions I talked about earlier. And then we have a third one we're going to add, which is photophosphorylation. This occurs in plants, algae, and cyanobacteria. So in substrate level phosphorylation, we have the direct transfer of a phosphate group by an enzyme to an ADP. In oxidative phosphorylation, we're using that electrochemical gradient, or the process known as chemiosmosis, to drive ATP synthase. And then in photophosphorylation, we're going to use light energy. So light energy is going to drive chemiosmosis, as opposed to, it's an electron transport chain, and light energy is going to be used to derive electrons and protons for chemiosmosis. And we'll talk about photophosphorylation in another lecture. So we're going to start in lecture 4. We will start with glucose metabolism, and we will go through the different stages of glucose breakdown and oxidation. See you in the next one.